Hello, this is Kimberly Glass, and I'm really excited today to talk to you about gene regulatory networks, which is an area that is very near and dear to my heart. It's an area of active research for me. Before we begin, I wanted to talk to you a little bit about some basic biology to make sure we all understand the importance of gene regulatory networks. The first thing to really understand before we dive into what a gene regulatory network is that biological functions is encoded in genes. The human DNA contains approximately 25,000 protein coding genes. Some genes contain information on how to make a special type of protein called a transcription factor. Right before a gene is a region of DNA called a promoter region. This enables transcription factors to control or regulate the behavior of a gene. So if you look at the structure of a gene, you have a start of a gene, end of a gene. That gene encodes a transcription factor, which can then go and bind to a promoter regulated gene. How transcription factors regulate genes influences how a cell behaves, and this is what ultimately causes biological functions in the cell. So why study gene regulatory networks? Well, we want to study gene regulatory networks because of this notion of biological processes. Biological processes are not driven by individual genes or proteins. Instead, they're driven by multiple connections linking together those genes, proteins, and other molecules. One way to model biological processes is through a gene regulatory network. In this type of network, nodes are genes. Some of these genes encode transcription factors. Edges represent relationships between the genes, and more importantly, edges are directed to indicate causality. One thing that's really important in the context of gene regulatory network modeling is that we need to compare gene regulatory networks. And by comparing these networks, we can identify alterations in biological processes that may underline differences between different biological states. So how do we get a gene regulatory network? Well, biological networks, and in particular gene regulatory networks, are not known a priori. Instead, they are created using genomic data. How we construct a gene regulatory network will often depend on what data we have and what we want the edges in the network to represent. It influences the method we use to construct the network. The meaning of the edges in a reconstructed network depend on the method used to generate those edges. It's kind of the flip, right? And it's going to ultimately influence how we interpret the interactions in a network. So before I talk to you about how we get gene regulatory network, I wanted to give a 30,000-foot view on gene regulatory mechanisms. It's a bit of an aside. My background is in physics, so this is a little bit of balls and strings, but I think it very well encompasses all the complex structure that goes on in gene regulation. So you can kind of think of the DNA as a long string inside the cell, right? Along that string, you may have a gene, and this gene starts, as we already noted, at the transcriptional start site. Along the DNA, you may have places where transcription factors can bind. This can be measured either in vivo using things like ChIP-seq data, or using computational approaches to try to predict potential transcription factor binding sites. Transcription factors can go and they can bind to these binding sites, but they don't act alone, right? Transcription factors are themselves proteins, and they can dimerize through what are known as protein-protein interactions. Along the DNA, you also have things such as genetic variants, where the DNA sequence is different in a particular individual compared to the rest of the population. You also have places where individual CG sites are methylated, and this is the notion of CpG methylation, and this impacts the DNA structure. Speaking of structure, you also have things such as chromatin state, chromatin domains, 3D chromatin structure, where the DNA wraps in itself, making itself more open and more closed, enabling transcription factors to bind and regulate genes. The ultimate outcome of this process is mRNA, gene expression. But this is not the end of the story. MicroRNAs can actually go and bind microRNA binding sites along mRNA and degrade this. Ultimately, the mRNA is then translated into protein. And you look at all this, and this is a lot of complex mechanisms. But at the end of the day, in a gene regulatory network, what we want to do is we want to deduce this to its simplest form. We want to take our two transcription factors and say we know that they do something to regulate their downstream target gene and influence its behavior. And we want to map all these connections out. So to get started, I think it's useful to focus on three basic mechanisms of gene regulation. You can start with something like transcription factors, right? And these transcription factors do not act in isolation. Instead, they work together and they form protein complexes. These protein complexes can be measured through protein-protein interactions. And in fact, protein-protein interaction networks by themselves were a large area of study. These protein complexes aren't just floating around the cell. Instead, they come and they bind upstream of the gene through protein DNA interactions. They bind through the promoter region and recruit RNA polymerase. RNA polymerase then goes along the gene and creates an mRNA product, or gene expression. This mRNA is further translated into a protein, and this protein may recruit other proteins to form a separate complex. This goes off to regulate other genes. These are three different sources of biological data, all representing the same underlying gene regulatory process. And ultimately, we want to model a gene regulatory network of this process to understand the structure of that network and to understand biological processes. Early approaches to model gene regulatory networks were largely based on gene expression data. The general approach is as follows. You have some sort of gene expression data where you have conditions on the rows, genes on the columns, for example, and use this to calculate some sort of similarity matrix. For example, pairwise Pearson correlation, mutual information. You can then threshold this matrix to define an adjacency matrix. Now, of course, this is a gene by gene matrix. We know that not all genes encode transcription factors. And so what's commonly done is that the rows of this matrix that refer to genes that do not encode transcription factors have been removed. Therefore, we no longer include non-TF gene interactions in this network. Now, there are many different types of gene co-expression network approaches. I only mentioned three here, and I mentioned them because they've been along for a very, very long time and are very, very highly cited and are still used commonly in benchmarking of new approaches. WGCNA stands for Weighted Gene Co-expression Network Analysis. The key idea of WGCNA is to take a Pearson correlation network, but then scale that Pearson correlation, this R-squared value, to a power. This allows the network to approximate a scale-free topology, which, as we know, is important for network structure in many common biological networks. CLR, or actually let's go ARachNE next. So ARachNE, which was probably next developed, stands for the Algorithm for Reconstruction of Accurate Cellular Networks. The key idea of ARachNE was to remove indirect interactions. It's based on the concept of mutual information. Basically, the idea is if gene 1 has a high level of correlation with gene 2, or mutual interaction, and gene 2 has a high level correlation of gene 3, and gene 3 and gene 1 also have a high level of correlation, then one of these three is probably an indirect interaction. And it used the triangle inequality to basically remove indirect interactions. Now, the problem with this approach is it removes all loops in the network, but the strength of the approach is it did get rid of many indirect interactions. Another commonly cited approach is CLR, which stands for Context Likelihood of Relatedness. The basic idea of this approach was to normalize each gene pair against a background. This also started based on mutual information and computed a mutual information matrix. But then instead of using that raw value, it took the Z-score of all the genes, mutual information across all the other pairwise interactions, gene I's mutual information against all pairwise interactions, and basically calculated a scaled version against these two backgrounds. This basically allowed every single gene to have at least one estimated edge, because some genes just had much noisier gene expression. It was harder to find mutual information of those genes. Now, as we all know, correlation is not causation, right? So up here on the upper left, I've illustrated what the expression of gene A compared to the expression of gene B might look like in a very simple toy example. Now, this type of co-expression can occur when one gene regulates another. For example, A regulates B or B regulates A, and that was the underlying principle of many of the algorithms that you just saw. However, this same pattern is observed when two genes are targeted by the same regulator. In this case, the transcription factor is expressed, but it's not until sometime later, after that mRNA has been translated into a protein and gone and maybe formed a protein complex, etc., that A and B are co-expressed together. Now, both of these are represent the same way in a co-expression network, right? Both these regulatory mechanisms are shown the same way, but it's impossible to really tell which one's going on. And this leads to many, many false positives and many, many false negatives when building a gene regulatory network just based on correlation types of data, whether or not it's scaled or otherwise. So how can we actually map direct transcription factor gene interactions? Is there a better way than using gene expression data? And the answer is yes, there are other ways to do it. So for example, one could choose to do it experimentally using something like ChIP-seq data. This will tell you all the locations of transcription factors bound in the DNA. You can then determine whether or not the transcription factor is actually binding to a promoter region, and you can deduce or estimate that because the transcription factor is bound to the promoter region of a gene, the transcription factor may regulate the gene. Now, the limitation is this is very expensive. You have to do a separate experiment for every single transcription factor. There are also a very limited number of antibodies. The experimental protocol does not work for every single transcription factor. And finally, binding does not necessarily equate to function. A transcription factor can bind to the regulatory region of the gene, but then do nothing else. It can just sit there and not actually cause the gene to do anything. The other way to build this map is through computational approaches. So for example, taking the known binding site of a transcription factor and scanning the DNA for instances of where that transcription factor might bind. If that transcription factor has a potential binding site in a gene, you can hypothesize that the transcription factor might regulate the gene. Now, there are still limitations. For example, we only know the recognition sequences for about 50% of transcription factors. So it's very prone to false positives. And more importantly, it's not context specific. Every single cell in our body has the exact same DNA sequence. But as we all know, the biological processes in the heart are not the same as the lung, are not the same as the liver. However, this is a very cheap approach. So traditionally, in constructing a gene regulatory network, a combination is generally taken where you combine computational predictions, such as those inferred on the right, with other types of omics data, such as gene expression. However, this is still very difficult to do and requires developing a real understanding of the regulatory mechanisms and caveats of different types of omics data. So this leads us to many advanced network methods for gene regulatory network inference. And believe me, there are many. I'm not going to touch on them all here. There are many methods for inferring gene regulatory networks. Almost all of them use gene expression data, and many of them incorporate other types of data, such as the types I mentioned earlier. Importantly, though, most perform very poorly in higher order organisms. This is a paper from Nature Methods in 2012, and I know it's about 10 years old, but I still think it greatly illustrates the problem with the field. In this case, they took a bunch of different methods for inferring gene regulatory networks. They benchmarked it against in silico data, in E. coli data, and in yeast. And you can see that when the underlying network structure is known for sure, such as in silico data, the methods perform reasonably well. The method's accuracy goes way down for something like E. coli, and then becomes almost impossible to discern for something as quote-unquote simple as yeast. Nevertheless, here are a handful of approaches that are again commonly used, commonly referenced, and commonly benchmarked against. Inferolator is an approach that uses linear regression, L1 shrinkage, and lasso to infer a gene regulatory network. GenieE3 uses tree-based ensemble methods to combine predicted transcription factor gene interactions with gene expression information and infer a gene regulatory network. And PANDA is a method developed by my group that integrates multiple different types of networks using message passing and to infer a gene regulatory network. So digging things a little bit more, how does PANDA work? The motivation behind PANDA is as follows. Multiple omic types of data are often very noisy, filled with both false positives and false negatives. However, the goal of network analysis is to uncover broad patterns in data, not individual relationships. So it's okay to recognize that there's noise in these data. So how does PANDA work? PANDA takes in 3 different types of network relationships: protein-protein interaction data, protein-gene interaction data, and gene-gene co-expression information. It then passes information between these 3 networks to find synergistic relationships between them, common structures, highlighting elements, for example, between pairs of genes that have a common upstream transcription factor, or pairs of transcription factors that share downstream target genes. These are some PANDA applications, which I don't have time to get in today, but just to kind of illustrate that the approach has been widely used in many different types of applications. And context and diseases. So we mentioned gene expression data. I mentioned some transcription factor binding data, but what about other omics type of data? Omics data. It's all very important for gene regulation. And in particular, I'd like to talk about epigenetic data, things like DNA methylation, chromatin state, and 3D chromatin structure. So what is epigenetic data? Epigenetic data tells us something about the physical structure of DNA. And in particular, epigenetics refers to genomic information that doesn't involve changes to the DNA sequence, right? Things like histones, nucleosomes, chromatin structure, things like that. So how does DNA structure impact gene regulation? Let's go back to our basic approach. We have our transcription factors that form a protein complex. In the context of epigenetic data, you can have something like the DNA wound around a nucleosome. This makes the DNA inaccessible, unable to be regulated if the promoter region is there. In this case, the protein complex may not bind to the DNA, RNA is unable to be recruited, and the gene is not expressed. And this is how epigenetic data impact gene regulation. So methods to derive gene regulatory networks using epigenetic data generally take the following approach. They look along the DNA for potential binding sites for a transcription factor, right? So for example, these blue boxes indicate the location of a transcription factor's motif or potential binding sequence. And you can see if the binding site of the transcription factor is in the regulatory region of a gene, you can draw an edge between that gene and the transcription factor, right? That edge here. If you have something like open chromatin data, say DNA hypersensitivity, ATAC-seq data, or histone modification data, you can actually use that to prune out edges in the network. This is done by pruning out motifs that are not in areas of open chromatin and only including links from transcription factors to gene where the motif of that transcription factor is found in the gene's promoter region and that motif is also in an area of open chromatin. 3 different papers that have used this basic approach, including this paper from Cell by Neff et al. In 2012. They basically use this exact approach, except they had DNase hypersensitivity data. And over here is an illustration from their paper where they looked at open chromatin in 6 different types of cell lines and built gene regulatory networks by filtering out transcription factor motifs. TEPIC has a similar idea. It combines transcription factor binding affinities based on epigenetic data and annotation data, gene expression data, to try to infer a gene regulatory network. And SPIDER, illustrated here on the bottom, is an extension of PANDA that basically prunes out relationships that are not in open chromatin, and it works by identifying consistent structures in this pruned network using message passing. So that's how you build gene regulatory networks. But how are these networks used to study disease? So you can think about in the context of a network, you might want to have a normal tissue network and compare it to a diseased tissue network. By comparing these networks, for example, by seeing edges that are lost and edges that are gained, you may be able to infer something important about disease. However, diseases typically aren't simple. You may have, for example, 2 different subtypes of the disease, and thus you're often not just comparing a normal versus diseased network. You're often comparing networks for different subtypes of the disease. So this leads us to common approaches to look at differential network analysis. These are a couple different papers that illustrate how this approach has been done. On the left, this is an approach I mentioned during the lecture yesterday, comparing an angiogenic versus a non-angiogenic network. Edges that are unique to the angiogenic network are shown in red. Those are non-angiogenic are shown in blue, and you can see some shared and distinct structures between them. On the right is an example of an analysis where there were different subtypes of PDAC. And this particular paper by Des Weigel, they looked at differences in gene targeting patterns in the network, gene co-expression networks, just based on gene expression data, and differential expression patterns. And they saw that different subtypes were associated with different types of biological processes, with the largest processes happening when they compared differential targeting, essentially differential degree of networks across the different subtypes. It is important to note that these differences could be more than pairwise. This, for example, is a paper that came out in 2021, where we looked at differences between different networks from different tissues based on GTEx. And we saw that different types of biological processes were associated with different tissues. And we analyzed 38 different tissues, making a very, very complex map between biological processes that were differentially targeted in these tissues. Now, this is important because this leads us to the notion of using networks for precision medicine, right? We're in a course on network medicine. How can we use networks for precision medicine? I earlier talked about two different network subtypes. For example, you can imagine that Sophia and Ketcha have two different types of subtypes of the disease. But of course, every single person is different. You might have different people that walk into the clinic. They vary based on their age, their BMI, their history, whether they smoked, their dietary patterns, etc. And all these things can influence their individual gene regulatory network. So how can we get gene regulatory networks for individual people, especially when these networks are commonly built using gene expression data? So most approaches for modeling gene regulatory networks follow the same basic principle. Almost every single approach I mentioned earlier follows this method. You take something like gene expression data, where you have genes measured across multiple samples. You may or may not integrate that with other types of omic data, such as transcription factor binding data or epigenetic data. And this gives you one network for an entire population. Now, this network represents information from all of the input samples, even if those samples represent a diverse set of conditions or people. So what we want to do is we want to decompose what we can refer to as the aggregate network into networks corresponding to each of the individual input samples. Now, there are two main approaches that were developed for this about a decade ago. The SSN approach, the sample-specific Pearson correlation network approach, is illustrated here on the left. And the Linus approach that was developed by my research group is illustrated on the right. Both of these approaches have the same basic idea. The idea is to infer a network with and without a sample and use the difference to estimate the sample-specific network. Since this is the same basic underlying approach, I'm going to dig into a little bit and how we do this in the context of Linus. So the motivation behind Linus is as follows. Estimating network relationships for multiple omic data often requires combining information from many different samples. But these samples are often obtained from a diverse set of individuals, each with their own set of clinical traits. So for example, when you apply something like the PANDA algorithm, you'll get one network that represents all the samples. So how does Linus work? Linus says that instead of estimating one network with all the samples, estimate one network with all the samples and one network using all samples except for one. Then apply the following equation, which I don't need to get into here. It's the derivation of the paper. And basically, this equation represents the difference between these two inferred networks, multiplied by a scale, adding back in a background. And if you do this for each individual sample, you can actually infer a network corresponding to each of these input samples. How does one use Linus in practice? I illustrated this in the case of PANDA, but Linus is highly generalizable. So for example, in the context of Linus, you might have a set of input data, which corresponds to n different samples. For step one, you apply a network reconstruction approach of your choice, be it PANDA, Gini3, a basic Pearson correlation, mutual information, etc., and use that to construct one aggregate network. For the next step, apply that exact same approach, except for apply it to all except for one input sample, sample of interest, sample Q. Then apply the Linus equation, and this will allow you to infer the network for sample Q. By repeating again and again and again, one for each sample in the input population, you can infer a set of corresponding sample-specific networks. We've also applied Linus to many, many different types of omics data and many different types of context. I've listed them all here in case you want to dig into any of these in particular, and also to understand how this might be used in different types of contexts. But I'm not going to go into them in detail here. I think it's important to mention that there are other new and emerging methods for sample-specific network inference. I mentioned the SSN method and the Linus method because those were some of the first methods developed and really formed the fundamental groundwork for this particular field. However, there have been other methods that have since been developed. So for example, the cell-specific network method is a follow-up method from the same group that developed the SSN method. This was developed in the context of single-cell data and uses essentially a non-linear approach. You can see in their little illustration here, they can find U-shapes or line shapes in the underlying data. And they use this to infer networks where they call individual cells. Just 2 years ago, the SWEET method was developed, and it basically borrowed a lot of mathematics from the Linus approach, and it changed an upstream scale factor purportedly to deal with differences in subpopulation weights. And just last year, the Bonobo method was published in Genome Research, and this uses basically a Bayesian approach to infer an individual sample's contribution to an overall aggregate network structure. Importantly, all three of these methods, the SSN method, the SWEET method, and the Bonobo method, have mathematics that is specifically derived in the context of Pearson correlation. And so when applying these methods, it's important to understand that the sample-specific network being inferred is a sample-specific Pearson correlation network, right? You can see that illustrated here in some of the mathematics that they refer to. Linus can similarly be applied to Pearson correlation and thus is comparable to these methods. But as I already mentioned, it's very generalizable, and it can also be applied to other methods such as mutual information, CLR, Panda, etc. If you're interested in single sample network modeling, I'm referencing here 2 additional papers, one from an outside group that I have no affiliation with, but I think did an excellent job comparing many of the existing approaches and benchmarking them in the context of oncology, and another commentary article written by some people in my group, Margarito Demarzio and Marika Kudra, a longtime collaborator. So to summarize what we've covered today on gene regulatory networks, gene regulatory networks provide a framework for modeling and analyzing biological processes. Despite many methods, regulatory networks are very difficult to infer. And even though it is an area that I have much interest in, I'm one of the first ones to tell you that we should not actually think about the networks as correct, but rather guiding principles for downstream hypotheses and follow-up experiments and validation. It is critical, but very challenging to effectively integrate multiple types of omics data into gene regulatory network models. I touched on transcriptomics data and epigenetics data today, but I didn't actually talk about some of the other omics data I mentioned, such as protein expression data, microRNA expression data, etc. All of these are very, very critical when modeling gene regulatory networks, but it's still hard to understand how to really get them into the network model in a concrete way. And finally, I mentioned the notion of sample-specific networks, and these approaches enable us to directly relate network features with clinical characteristics and phenotype. By inferring an individual sample network, one for each patient in the population, we can understand how network features change as a function of something like BMI, case control status, gender, etc. And it's really important for really connecting the models that are used to infer gene regulatory networks with many of the clinical characteristics that we encounter in the context of medicine. So with that, I'd like to thank you for your time, and I look forward to your questions at the panel discussion tomorrow. Thank you.